Hi, this is Mr. McLean. This is our second video in the Chapter 20 Biotechnology section. And the topics that we're going to go over today are going to focus on complementary DNA or cDNA, as well as PCR and gel electrophoresis. So to start us off, one of the things that is often made for being able to identify or see how certain areas of a genome are being expressed is to take a look at complementary DNA. And basically what that's going to do is it's only going to focus on mRNAs. So if we think about our DNA as having lots and lots of genes that are not expressed, and only those genes that are eventually going to form messenger RNA are going to be part of that process of expression, we're focusing exclusively on genes that are going to be expressed in different cells. So if we start here, one of the things we're going to notice is that we're taking mRNAs, which are obviously going to be the products of the DNA that we're looking at. So once we've collected the messenger RNA, we're going to have to convert that messenger RNA back into a form of DNA. So the enzyme that's necessary for that, and we're going to talk later on about how this enzyme is critical in some of our analysis that's going on today, is called reverse transcriptase. And reverse transcriptase allows for a reverse transcription. So we're going to take RNA and convert that into DNA, which is not our typical protocol. So if we take a look at this, here's my messenger RNA sequence, and the primer is going to be a collection of T's that will match up with the poly A tail, and then we're going to be using the reverse transcription moving in this direction towards the growing 3' like we always have. And what you're going to end up with is a sequence that's actually going to be single-stranded DNA that's based on the messenger RNA code. And then from there, this acts as our template to build the second strand of the DNA. So they're going to add traditional DNA polymerase, and at the end, you're going to end up with what's known as complementary DNA. Now, what's unique about cDNA is that it does not include any of the non-coding regions. So if we're specifically trying to focus in on the expression portion of a particular genome or a certain gene in study, this would be a technique that would be useful. So it's called complementary DNA. Now, one of the things that a lot of times we have to be able to do is we have to be able to find a gene of interest. So for example, if there's a certain known target sequence that we're looking for, whether it's in an analysis of a different test or some other sample that we're trying to identify a particular sequence, what we can do is we can create what's called a probe. Okay, so this probe right here is looking for this complementary sequence. So the desired gene sequence is listed up here in the black. So we've created a single-stranded probe that happens to be labeled with some form of radioactive label so that we'll be able to identify when hybridization occurs and we pair these two things together. So the idea is that you can look at a lot of different genes in a particular sample and try to identify specifically the genes of interest. So one of the ways in which they would do that is as we were storing our different clones, we showed you in a previous video that they could often be stored in these microwell plates. And the microwell plates are going to be exposed to this nylon membrane. Okay, and the nylon membrane is going to absorb to a certain extent some of the DNA that's stored in these different plates. We're then going to put it inside of what looks to be like a Ziploc bag, and within there will be a collection of these radioactively labeled probes. So the sequence that we're looking for, once again, was this sequence in black. So anywhere where that sequence exists, our DNA probe is going to hybridize to that. It's going to partner up with it in a complementary fashion. And because this probe has a radioactive labeling associated with it, we can use something like a film, a piece of photographic film, which will create a exposure in that particular area. And that's going to allow us to go through there and be able to actually identify which of these wells actually has the gene of interest. So from our observation, we can see it's right here. But when you actually run the test on the film, it'll point out where the specific location is at, and then you can target that. Now, that's definitely a piece of technology that's being used today. This next section is really critical. This is a tool for amplifying DNA. And this was developed in the 1980s here in the United States. And if you watch any of the coverage related to some of the testing for the coronavirus, one of the things that you're going to hear quite a bit about is PCR, which stands for polymerase chain reaction. And basically what this is going to do is it's going to allow us to make multiple copies of a given sequence. So whether it's a DNA sequence or an RNA sequence, this protocol will work for us. And in the case of what we're looking at regarding coronavirus, it is an RNA-based virus. So what they're going to actually have to incorporate is another step where they have reverse transcription and then PCR. Okay, so we'll take you through the basic PCR protocol. So what we're going to find is there's three main steps to this, and it's a cycling. So you're going to have a heating section, and you're going to have a cooling section, and then you're going to have a time where it's going to be able to replicate. Now, one of the major discoveries that made this protocol possible was the identification of a DNA polymerase known as Taq polymerase, which is very heat stable. So typically when you're thinking about something that's enzyme based, there's going to be a limit to what temperatures it can tolerate before you start to see breakdown and changes. In this particular scenario, this was derived from some thermophilic bacteria that can tolerate and really enjoy high temperatures. So let's suppose that we had a section of DNA within our genomic sample, and we wanted to amplify this particular sequence. So we'd cut it and we'd take a sequence of this, which is our target sequence right here. So in the first stage of the cycling, it's classified as the denaturing cycle. So when we think about denaturing, we're talking about breaking something down. That's why it's so critical that we use the Taq polymerase, because it can tolerate the heat. When we separate these strands by heat, we then use step 2, which is the annealing process. And the annealing process is going to use primers. So we're going to have our specific primers incorporated in there. Now, as I said, if you're looking at RNA, you'll have to use a reverse transcriptase capability. And the primers, as well as the polymerase at this point, will allow for the extension stage where you're going to build new nucleotides. So essentially what this does is every cycle you have a duplication of your quantity of your nucleic acid. So after multiple cycles, you can build a much, much larger sample. So if we're looking for a specific sequence, for example, the sequence that would be found in a sample of somebody who is testing positive for coronavirus, or even if they have a negative sample, part of what we're trying to do is we're trying to build up a quantity of that RNA sequencing so that we can kind of go through there and make sure that we get a clear analysis of whether that person actually has that issue or not. So this is a technique that is, it's been around since the 1980s, but it's clearly very critical today. And there's a little video in here that talks about some of the different techniques associated with PCR. All right. And then the other thing I really wanted to focus on in this chapter is something called gel electrophoresis. And gel electrophoresis is a technique that allows us to separate different molecules based on things such as size and electrical charge and some of the other physical properties. In this particular case, we're going to be talking about gel electrophoresis as a way to separate nucleic acids. So they're going to have to use a special gel, and with this special agarose gel, it acts as a molecular sieve, if you want to think of it that way. And as the molecules are going to move based on an electrical charge, what we're going to find is that molecules that tend to be smaller are going to get less resistance, and they're going to move a further distance. And the molecules that are going to be larger are going to feel a greater resistance. So if we look at this picture right here, in fact, let's zoom in on this one right here, what we're going to see is these are different DNA samples that we want to compare. So let's say for a forensic investigation, maybe in this first tube right here, we have DNA that was collected from a crime scene, and maybe we have two samples that were collected from two prospective, assailants in this particular case right here. And as we kind of go through this, our suspect DNA is going to be compared against the DNA that was recovered from the scene. We put them into these wells, and when we set the gel up, you use what's called a comb. So it's a little plastic device. It looks like a comb, and it creates these little depressions inside the gel. So we insert the liquid DNA mixture as well as with a buffer solution in here. And the special gel, like I said, it's sitting in this chamber. It's an electrophoresis chamber. It has buffer solution, and it has a power source that connects to it. So the power source runs a current from this end towards this end, and it runs from negative to positive. And the idea is the DNA molecule will progress towards the positive end of the chamber because of the charge associated with the molecule. So the slight negative charge is going to be attracted towards the positive. And if we look at this picture right here, what we're going to see is that the fragments are going to the small fragments of the DNA, which were cut in advance. So I guess I should go back and explain that. We cut these up with different restriction enzymes, and as those fragments are going to move through here, what we're going to notice is that they're going to break down and deposit in these different areas depending upon the size. Now, we then go through there and we can use a blacklight or some other visualization technique, and this will create basically a map of the the DNA sequences that were cut up. So if you had two individuals or two samples that were from the same individual and they were cut in the same regions using the same restriction enzymes, what you would see is a very identical pattern. If you have patterns that are non-identical, what that's telling you is that their DNA sequences were different, and therefore it wasn't from the same person. Now, how we would go through that, we could also use that for other applications such as in medical diagnostics. So it's called restriction fragment analysis. And what they can do is they can look at comparing different restriction sites. So in this case right here, they're comparing somebody with sickle cell versus a normal scenario. So a person that has sickle cell, what they're going to find is that they don't have the exact same sequence as the normal hemoglobin. So that change in that sequence, when we use these restriction enzymes, this fragment doesn't cut where it normally would cut. So you notice that the size of this fragment is 376, whereas in persons that don't have this, they have a cut, so they have a 175 and a 201, and as a result of that, what that's going to do is that's going to give us a bigger fragment. When we go over here and we compare this with the gel electrophoresis, what we're going to see is the person with a normal allele, they're going to have a large fragment, that's this portion right here that's going to band out, and then they're going to have a 201, which is bigger than the 175, and the 175 is going to move down to here. When we look at an individual that has sickle cell, they're going to have that large fragment, and they're also going to have the 376 base pair fragment that's going to kind of target right here, and that would give us some evidence that those individuals have a unique DNA sequencing. So hopefully that helps. We'll take a look at some other advanced techniques in another video.